Good afternoon, hello and welcome to the Department of Biochemistry at the University of Cambridge in the UK. I'm Dr. Reese Grant and I am the Science Communications Specialist here in the Biochemistry Department, shared also with the Cell and Molecular Biology Programme at the Cancer Research UK Cambridge Centre at Cambridge University. So I'm extremely pleased to welcome you to our departments this afternoon for this live talk as part of the 2021 Cambridge Festival, which aims to tell you about some of the exciting science that's being undertaken by researchers at the University of Cambridge. So for this talk, I'm joined by one of those researchers, Dr. Ross Waller, who is a research group leader in our department at the University of Cambridge. And today, Ross is going to be talking to us about how life's diversity is founded on the partnerships between different cells, such as the partnership between corals and algae that is essential for reef health. I'm also joined, as you can see, by Latika Sugamba, who is part of our department's secretariat team. And Latika is going to be helping me out this afternoon behind the scenes with the running of this talk. Before we get on to any of our exciting science though, the first thing that I'd like to ask you all to please do is to click the subscribe button down below to follow our YouTube channel, because we have loads more talks coming up this week for the Cambridge Festival, and we're planning on bringing you even more live content from our department in the near future. I'd also like to encourage all of you to please check out our other social media channels on Twitter, Facebook, Instagram, LinkedIn, and so on, using our handle @CamBiochem. So that's CAMBIOCHEM, because we use these channels to tell you about all of the news, the research, the teaching, and everything really that goes on in our department. I also just want to mention that after this afternoon's talk, Ross, Latika, and myself will be having a live question and answer session. So if you have any questions on Ross's talk, or even if you just want to ask us more general questions, for example, about what life is like at Cambridge University, please post your question in the live chat on YouTube, and then the three of us will stay for about 15 minutes after Ross's talk to answer as many of your questions as we can get through. If you do want to participate in the Q&A session, you don't need to worry about being on camera, just in case you're munching your way through your lunch or anything like that at the moment, because you're not going to be seen and you're not going to be heard. It's just going to be me reading your questions out on your behalf anonymously. So, as I mentioned, this afternoon's talk is going to be given by Dr. Ross Waller, who is a research group leader in our department at the University of Cambridge. But Ross didn't begin his career in Cambridge. In fact, Ross began his career in Australia, where he completed his PhD at the University of Melbourne, working on the newly discovered remnant plastid endosymbionts of the parasites Plasmodium and Toxoplasma. Ross then undertook postdoctoral training in Melbourne, where he worked on Leishmania, which is another human pathogen, before he then moved to Vancouver in Canada for 2 years to work on the cellular and molecular evolution of diverse eukaryotes. In 2005, Ross returned to Australia, where he joined the faculty of the School of Botany at the University of Melbourne. And then in 2013, Ross relocated to join our department in Cambridge in the UK. Ross's research now continues to focus on how microbial organisms form partnerships, or symbioses, with animals and other organisms, either as pathogens that cause disease, or as beneficial partners that are necessary for healthy ecosystems and environments. So with that, I'd like to hand over to Ross to start this afternoon's talk. So thank you very much, Ross.
Good. Thanks very much, Reese, and thanks everybody for joining us today. I want to start with this beautiful image from NASA that focuses our attention on the oceans of the world, because here we can see colored in increasing shades of blue the photosynthetic activity that we find in the oceans. And you can see it's very mixed, but there's lots of areas of higher concentration, because the oceans are one of the important engines of life and of energy on the planet still. And in fact, a lot of the partnerships I'm going to talk about today actually started in the oceans. So this is a very good place, I think, for us to start this discussion. So I'm going to move to coral reefs. And here's a some images of coral reefs, wonderful ecosystems. They're in fact the most biologically diverse places on the planet. Coral reefs are up to 100 times more biologically diverse and rich as rainforests. So they really are incredibly important and incredibly useful parts of the environment. But it's not just the animal life that they sustain. You can see fish and other organisms in the background here, but they also form often these rocky, hard structures that form reefs that can protect coastlines. So here's an atoll on the bottom right-hand side. It's being protected from the wave action of the ocean, stopping the wave action so it doesn't wash this sandy island away. And coming from Australia, I know that thousands of kilometres of our coastline are protected by these coral barrier reefs. So it's not just in the ocean that coral reefs are important, they're also very important for land and maintaining our coastlines as well. The coral reefs are really a very good place for us to look at these cellular partnerships. They're a really classic example of cellular partnerships, but in fact, there's probably more to these partnerships than you might have already guessed or realised. So I want to start with a perspective that we often have when we think about life. We often think about organisms as individual entities. So here, these dolls here might represent different animals in this marine environment. Perhaps the largest one is a whale, the next one's a shark, a fish, maybe some crabs, some crustaceans, and then some microbes. And often we think about these as individual organisms in their own right, as discrete creatures. But Russian dolls tell us that, in fact, what's on the surface is not always the whole story. And if you're used to playing with Russian dolls or you're familiar with them, you know that there's many layers of other things inside that you don't see at first glance. And I think this is actually a very good picture of life, in fact. And we're going to explore corals as a nice example of how these partnerships and multiple partnerships actually can create the modern life that we know and are familiar with. So this is really a story about cellular partnerships. And in order to understand the cellular partnerships that are important for corals, we actually need to go back in time. In fact, we need to go way back. We're going to go back something like 2.7 billion years ago. And we're going back to some of the early ancestors of corals and also the early ancestors of indeed us. So this is a representation here of our ancient ancestors called an Asgardian. So here we can see actually some modern pictures of Asgardia. So these are our very ancient relatives, and they're made up of single-celled organisms. So a cell is, of course, the basic unit of life, and we're made up of multiple cells, but most life actually is still made up of single-celled organisms. And these are good examples. Here's Asgardia. So this name's a bit of a mouthful, but archaean basically refers to the fact that these ancient types of organisms, they're actually called Asgard because Asgard, in fact, is the mythical location of Norse gods like Thor here, the god of thunder with his magical hammer here. So these organisms have actually only been recently rediscovered about 10 or 15 years ago off the coast of Norway in deep ocean sediments. And you might imagine with a name like this, the Asgardia, that they'll be quite powerful, quite impressive organisms. But actually it's anything but the truth. These are some of the weakest, wimpiest of the cells out there these days. And I'll tell you why. It's because like us, Asgardia need to get their food from the environment. They need to eat things to get energy. And we certainly eat things that are rich in molecules like glucose. But there's a real problem with using glucose as your energy source. And that's because there's so much energy in glucose that the cell usually can't do much with that if it were to release all that energy at once. Using glucose in a cell is a little bit like trying to light a candle with a stick of dynamite. It might be that Thor lights his candles this way, but you can imagine if we tried to do that, we would blow the candle in ourselves to smithereens. And this is the problem with glucose. There's too much energy in glucose for most of the cellular processes that have to take place. In fact, what we need is something the equivalent of a match to light the candle. And the molecular equivalent of a match is a molecule called ATP. So cells have got this challenge. They need to turn dynamite into matches so that they can spread that energy over lots of processes and use it in a very controlled way. So archaea can do this, but the problem is they're very inefficient. For every stick of dynamite or every glucose molecule, they only get a few matches or a few ATP molecules. And this really slows them down because there's only so much food in the environment. If you're not making the most of that food and the energy from that food, you're not going to be very competitive in the environment. So archaea effectively are these fairly powerless wimps, if you like, in the environment. They're still not very successful, and that's why we only find them in very obscure places like the deep sea sediments. However, there's another group of organisms, bacteria in fact, that have been much more clever in the technologies that they've developed. And a group here that I'll talk about, the alpha Proteobacteria, they learned how to make glucose into ATP or dynamite into matches much more efficiently. And in fact, alpha Proteobacteria get about 15 times as many ATP molecules per glucose as the Archaean does here. And then this is enormously useful. You can imagine it's like having this extra spending power from your glucose. You can do a lot more things. You can divide more quickly and grow more quickly and become much more active. So this is an advantage. And the Archaebacteria decided that this is something that they wanted to adopt. And they did that through a partnership. They basically made a partnership with a Proteobacterium, and the partnership is called an endosymbiosis in this case. So symbiosis, because it's two things living together, they're symbiotic, and endo refers to the fact that one of them is inside the other. So this is an endosymbiosis. And here we've now got the alpha Proteobacteria inside the Asgard Archaebacterium. And this indeed is a permanent symbiosis. We don't actually know exactly how the alpha Proteobacterium got inside. It might have been that the Archaean swallowed it up, or it might have been that the Proteobacteria burrowed its way inside this cell, or maybe they just lived closely enough together for long enough that one just grew over the other one. But one way or another, we find that in most, or certainly a lot of modern life, this symbiosis continues here. And we can see this in a compartment that we now call the mitochondrion. And here's some mitochondria in an electron micrograph here in a human cell. And they actually still look pretty much like bacteria, but this is now a permanent endosymbiosis. So this has been an incredibly successful partnership because now the Archaeobacteria has got the power of all of that energy it can get from glucose, and this has led to a huge radiation of life on Earth. And indeed, we're a product of this union. So here's us down here. We're just one of these groups. This group are now called the Eukaryotes that contain this bacterial symbiont as a mitochondrion. And as I say, this really drove a lot of the radiation and the evolution of complex life like us. And we still have these bacteria inside us. We're basically a partnership between two cells, the ancient Archaean relative and this endosymbiont. I want to introduce now another type of bacteria called a Cyanobacterium. And again, bacteria have been the masters of innovation. And these Cyanobacteria, they developed another process called photosynthesis. In fact, there are many forms of photosynthesis, but Cyanobacteria made the most efficient form. And this, of course, is the process of harvesting the energy from sunlight and converting that directly into food like sugars, like glucose. So this, you can see, is an enormous advantage. You don't have to go and find food in the environment. You can simply make it using sunlight. And a byproduct of this very efficient form of photosynthesis that Cyanobacteria invented is oxygen. It in fact is a gas that gets released in this process of harvesting the energy from sunlight. Now you might know of Cyanobacteria, they get a bit of a bad name. Often they're associated with algal blooms, and here's the Great Union Canal here that's full of a Cyanobacterium. They can even produce toxins and cause toxic algal blooms. But I want to change your attitude towards Cyanobacteria because we should be extremely grateful for them. Going back to that image that I showed before, this image of chlorophyll concentration, or effectively photosynthesis in the ocean, about half of the photosynthesis that takes place on the planet is in the oceans. Half of it is in the land, half of it is in the ocean. So half of the power that we're harvesting from the sun comes from the ocean. And actually, the Cyanobacteria is still a major part of that community that are making, that are responsible for photosynthesis in the ocean here. Now, it's worth thinking as well that this oxygen that's being made, we use up, the whole planet uses up the entire Earth supply of oxygen every 20 years. So we're relying on Cyanobacteria in the ocean, plants on the land to replace that oxygen. So every fourth breath you take has been made by Cyanobacteria in the last 20 years in the ocean. So you want to be, I think, pretty grateful for Cyanobacteria. But there's another reason I think we should be grateful for Cyanobacteria, or another important reason, and that's because by making oxygen, they really changed the atmosphere of the planet. There used to be almost no oxygen in the atmosphere. It was a very anoxic environment. And as the cyanobacteria developed photosynthesis and became more prolific, they changed the atmosphere of the ocean and the atmosphere. But this changed the chemistry of the ocean in a way that made all of the iron that used to be soluble as a ferrous type of molecule in the ocean, it made it a insoluble ferric form. It basically oxidized all of the iron in the ocean. It caused the iron to rust, and then it settled down to the bottom of the ocean and formed deposits that are basically now our iron ore that we can make metals from. So in fact, if it wasn't for cyanobacteria concentrating all that iron in these sediments, we wouldn't have developed technologies based on iron, because there wouldn't have been enough iron in rocks to be able to utilize that. So I'm going to give our little person down here an iron shell here, a suit of armor, just to remind us that we should be grateful for cyanobacteria for more than one reason here. Once again, we've got a fantastic technology. You don't need to eat, you don't have to find food, you can be photosynthetic. And some of the eukaryotes decided that this is a partnership they couldn't resist going into. So again here, we see a partnership, an endosymbiotic partnership with a eukaryote that already had a mitochondrion, so it's already got a Proteobacterium. And now we've got a cell with two cells inside of it. The chloroplast, we call it the chloroplast now, is this living cyanobacteria that continues on, and the mitochondrion as the alpha Proteobacterium. And again, this is an enormously powerful combination, because the chloroplast is making glucose, the mitochondrion can use that very efficiently to make as much ATP as possible. We've really got a very competitive, powerful cell here. And again, we see enormous radiation of life from this early event. We see all of the plants a product of this endosymbiosis. And indeed, if we look at these plant cells in a leaf here, you can still see the cyanobacteria as chloroplasts here. Each of these is the individual relic of that cyanobacterium symbiont. We also get beautiful algae in the oceans, some green algae here and aquatic lakes and so on. And all of the red seaweeds are also a product of this symbiosis. You might enjoy going to the ocean and fossicking for beautiful seaweeds like this, or you might just like to eat sushi, and sushi is wrapped in a red alga. Even though it looks green, it's actually a red alga. So again, an incredibly important event in the evolution of life, and an incredibly powerful partnership. So I'm going to put our red alga down there. We also call this a rhodophyte. Rhodo is red, and phyte basically just means a photosynthetic organism like a plant. So red algae, rhodophytes are going to be part of our ongoing story here. So I'm moving a little bit forward in time, about roughly 1.2 billion years ago. And now I want to introduce another eukaryote, another type of cell. So again, it's a derivative or a descendant of this organism back here. It's a eukaryote, it's got a mitochondrion. But like us, it needs to eat. It needs to find food in the environment. So this is a single-celled organism. It swims around with these little flagella looking for food, and it really enjoys eating other algae. Now, algae are a great food source for this organism. This is called a cryptophyte because it's got a crypt, or a small gullet there that it feeds on other cells through. So one of the types of cells that cryptophytes like to eat are actually red algae, single-celled red algae, not this multicellular one here. And here's a picture of a cryptophyte that has swallowed one of these red algal cells. And we can see that pretty clearly by the color there. It's now got that red pigmentation of red seaweeds. Once again, one of these cryptophytes decided that this is a relationship that's too good to give up. I don't want to just digest this food, but it maintained this as a permanent symbiont here. So now we've got a rhodophyte inside this cryptophyte here. So now we've got a combination of 4 cells making up this organism here. Cryptophytes, you might not have heard of them before. They're environmentally not very important. They are very common in coastal areas, very common in lakes and freshwater systems. But from an ecological perspective, they don't have a huge impact on the rest of life. But they have played this incredibly important evolutionary link in a chain that I'm going to continue to tell you about. So we'll pop our cryptophyte down there. Now I want to introduce you to another organism, another eukaryote, so another cell that basically had to find its food, but was using that food very efficiently with a mitochondrion. Again, this is another organism that's out in the environment looking for foodstuffs to eat, often swallowing whole other cells. And what happened here, you might have guessed this already, is that one of these cells swallowed up a cryptophyte and again made this a permanent symbiont. Photosynthesis was too good of a tool or a technology for this cell to be able to resist, and it's become a very efficient, stable endosymbiont again. Now, these are really important organisms. So these are called Okrophytes. So again, the color is indicated by ochre, so it's yellowy brown in color. And phyte basically is referring to it being another photosynthetic organism. And you'll be familiar with these, I'm sure, because these are the brown algae and the kelps, beautiful organisms that are forming incredibly important ecosystems around coastal areas. You can see here these kelp forests, great habitats for fish and all sorts of other organisms. These are multicellular okrophytes, but the more important okrophytes are the single-celled ones. And these are some beautiful cells here called diatons. And you can see, in fact, this diatom, it's got a shell made of glass. So this is an image of this cell looking from the outside. And this beautiful ornamentation here is made of glass, it's made of silicon. And there are all sorts of shapes of diatoms. You should have a look at some diatom examples with a good Google search. But these are important because, again, these are one of the major plankton species in the ocean that are photosynthetic. They're similar to cyanobacteria. They're probably responsible for about every 5th breath you take of oxygen. You can thank diatoms in the last 20 years for making that 5th breath possible. So again, really important ecological drivers in the ocean. Okay, so now we're going to go and look at another cell. And these, in fact, are some of my favorite cells. So this is, again, another eukaryote. You can see there's a common pattern here. This is a dinoflagellate, again, mitochondrion. These are beautiful organisms. And these images here, microscopic images here, show what these cells look like. They're single-celled organisms again, but they've got this beautiful sculpted forms. So these are just a treasure to look at and to explore. Now, a lot of these dinoflagellates, again, they're still feeding organisms. We call them micro-predators because they run around in the ocean predating on other single-celled organisms. Once again, you can see that this is a good recipe for a partnership. And in this case here, it's been an okrophyte that's been swallowed up by a dinoflagellate and has become a permanent symbiont. So here we've got a dinoflagellate down here. Again, we can see the pigments, the photosynthetic pigments of the okrophyte inside. And these organisms now have become photosynthetic as well, or at least some of them are. Again, dinoflagellates are incredibly important in the oceans. Maybe about every 7th breath or so, you should thank dinoflagellates for producing the oxygen for that breath. So you can see now we're actually really stacking up the number of cells that are all layered upon each other. And again, you can see that there's this pattern of cell partnerships that's going from one step to another. It's an ongoing progression. So finally, we're going to get to where I said we're going to arrive. We're going to talk about corals. And again, a coral is an animal. So it's actually like us. It's another eukaryote. And I'm showing you 2 pictures of corals here. Well, I've got coral here and a close relative of coral and an anemone. Now, you might be wondering why these corals look so white, because that's not what we expect of these corals. But what I'm showing you here is the coral by itself. What corals should look like is they should be colorful. They should be colorful like this coral reef that I'm showing with multiple beautiful shades of brown. Or here's an anemone here with those tentacles all colored nice and brown and red colors. These colors come from another symbiont. And in this case, it's a dinoflagellate that is in a partnership with the coral. So here we've got down the bottom right, you can see those dinoflagellate cells inside coral tissue. The white around it is coral tissue. There's no pigment there or very little pigment. And the color of the coral is coming mostly from the dinoflagellates. So again, this is an important partnership. And it's an unusual partnership because all of these other ones I've talked about so far are permanent partnerships, whereas this is not a permanent partnership, as we can see here by these bleached examples where the coral has lost its dinoflagellate partner. So what do the corals get in for the partnership with the dinoflagellate? What's the dinoflagellate get in from the partnership with the coral? It might surprise you, but corals and coral reefs grow in very food-limited environments. The lovely crystal clear blue seas you often associate with coral reefs, the clarity is because there's not much food and not much nutrient in that water. So corals are animals, they feed, but without much food, they would actually starve to death. And these bleached examples here, if they don't acquire new partners, new symbiotic partners from dinoflagellates, they'll die for sure. They have a partnership with dinoflagellates because dinoflagellates don't need food. They can make food from sunlight through their partnerships, through their endosymbionts back here. So dinoflagellates in the sun can make sugar that they share with both the dinoflagellate and with the coral or the anemone. But what do the dinoflagellates get out of it? Well, again, dinoflagellates need nutrients. A bit like plants, they need nutrients like nitrogen. And coral reefs, coral seas are often very poor in nitrogen. But nitrogen is a waste product from animals. We get rid of a lot of our nitrogen in our urine. So this is a source of nutrient for the dinoflagellate. And in this case here, the partnership is that dinoflagellate gets nutrients from its host, things like nitrogen, and in return, it photosynthesizes and gives sugars. So it's really a very good partnership between these two. I want to take a diversion here and tell you about another organism partnership. And this might surprise you a little bit, but it's very closely related to this dinoflagellate coral partnership. So I need another host organism here, and that host organism is us, as humans. And we've been going steadily through time, and these are approximations. It's hard to get accurate measures of these, but this is a good approximation. But basically, these are partnerships now that we can enter into every day. And I'm going to come back and look at the dinoflagellates here, because one of the relatives of dinoflagellates did an unusual thing. Rather than using this endosymbiont, this plastid or chloroplast here for photosynthesis, it actually lost the photosynthetic capacity of that organelle, that structure. And these relatives of dinoflagellates are called apicomplexans. So they're just the close relatives of dinoflagellates. So apicomplexans lost photosynthesis in this structure here, which is a really strange thing to do, because it went from being able to make food from sunlight to losing that ability. And now it needs to get food from some other organism to replace that. What you might imagine it's going to do is look for a good source of food. And in this case, it's decided that we're a good source of food. So this is an apicomplexan is another organism that's going to form a symbiosis with humans. It's actually going to invade our cells and go inside and actually get its food that it needs from inside of our cells. This probably sounds like not a very favorable partnership for us. And indeed it's not, because these are the partnerships that lead to diseases like malaria. So in this case, the apicomplexan here is Plasmodium, and we're its host. So here in this image here is human blood. So these are human blood cells, you can see, all squished together under the microscope. But inside the human blood here, inside one of our cells, is a Plasmodium parasite. So it's one of these apicomplexans. And then inside the apicomplexan, shown in green here, are all of these other compartments. So the apicomplexan has kept all of these other partnerships because they brought still a lot of important biotechnology and biochemistry to the cell. So we can actually see in this image here, this multiple layers of Russian dolls, if you like, with a host cell, the Plasmodium parasite, and then all of these other cells all compacted down into that little green dot there. So partnerships they can go both ways. They can be symbiotic, where we both get benefits. They can be mutualistic, like the coral partnership here, or they can be a little bit unbalanced in terms of that partnership, or even very unbalanced in the case of malaria, where in fact we lose from the relationship with the Plasmodium cell. It's a parasitic relationship in this case. But I want you to appreciate that this is the view that we often take when we think about organisms. We think about humans, we think about corals, but in fact, we're much more than that. If we peel away those layers, we see that there is multiple partnerships in each of these organisms, including us. I mean, we certainly even have a mitochondrion, but at certain times we'll have relationships with other organisms, and some of those can be beneficial, some of them can be detrimental, like the Plasmodium parasite there. So in biochemistry, we're studying all of these relationships. We're studying malaria and Plasmodium and how Plasmodium interacts with human cells, how it gets into human cells, how it exploits them, and how all of this evolutionary technology that the malaria parasite has carried along all this time, how that continues to contribute to the biology of Plasmodium. And by understanding these facets, we hope to be able to disrupt this partnership or this relationship. But we're also studying the partnership between corals and dinoflagellates. So again, this is a partnership that's not permanent. And if the sea gets too warm, if the sea gets too dirty with pollutants and so on, we know that we lose this partnership, and the dinoflagellate leaves the coral, and the coral eventually will die. And think about all of the habitat, all of the ecosystem, all of the structure on coastlines. It's at risk if those partnerships break down. So again, it's important to understand the relationship between dinoflagellates and corals, understand what makes that relationship and what sustains it, to be able to hopefully manage our environment as well as possible, so that we don't lead to this irreparable breakdown of these relationships. And we're also studying all of these ancient endosymbiotic events as well, because they're literally the history of life. And how those partnerships took place, how they have built on one another, is really the story of what modern biology is. So we need to look back in time as well to think about and learn about cells and what defines cells. So I've talked today about, or focused at least mostly on corals, but this is just the tip of the iceberg in terms of cellular partnerships. These partnerships are in all parts of the environment, in reefs, in forests, in other parts of the ocean, and also in agricultural settings. There are essential partnerships between agricultural plants and fungi and bacteria that again are just as important for the health of those environments. And it's also our urban environments as well. And that includes us. And we're learning more and more about the partnerships between our bodies and microorganisms and other organisms around. So really, we need to understand these cellular partnerships to really have a full appreciation of life. So thanks very much for listening. I hope you enjoyed that, and I hope you enjoy the rest of the festival. And if anyone wants to stick around for questions, I'll be happy to stay on.
Thank you very much, Ross. That was a lovely talk, and life really is a matryoshka doll. I mean, I've heard this analogy before, but seeing it on your slides, it's just kind of blown me away a little bit. So thank you very much. So we're now going to have our live question and answer session. So if you have any questions, if you could post these in the live chat on YouTube, and then I'll read out the questions for you anonymously on your behalf. And there is a 20-second delay though between us talking on Zoom and you hearing us on YouTube. So I hope you don't mind, but I'm going to ask the first question. So I was just wondering, when that initial Asgard Archaean formed the symbiosis with the alpha Proteobacterium, do we assume that the Asgard was faulty in some way? I mean, why didn't it just eat it? And I guess that's the same for each of those steps. Why not just eat the thing that's gone inside you?
That's a good question. And I think I'd probably also just point out that many of the archaea would have eaten the bacterium, or many of these relationships that I showed you. There's a lot of testing and a lot of trial and error that happens in biology. A lot of those relationships, particularly where one cell is eating the other, most of the time, what got eaten gets digested. But we see, and this is happening all the time in lots of modern examples as well, there are cases where digestion is slow for some reason. And if it's slow for a while, and that organism is still doing something biologically useful, like making more sugar, then there becomes a selective pressure to slow that digestion down. And we see evidence of that. So all of those possibilities would have happened. Some of the cells would have just eaten the organism and not realized what they're losing. But it only takes one one organism to eventually decide not to do that. And if that becomes so successful, it can go on and proliferate. So in the case of the archaea, we know that there are still those Asgard archaea out there. So there are many that didn't eat the cell or perhaps digested the cell, but there was at least one out there, and probably many more at the time, but at least one that maintained that symbiosis, and we just see the legacy of that because it was so successful.
Actually, there's a follow-up question from a member of the public on YouTube. So it's quite clear to see the benefits to the Asgardian archaea for the endosymbiosis. But what were the benefits of being part of that Asgardian from the Cyanobacteria or the alpha Proteobacterium's perspective?
Again, that's a good question, and it's easy for us again to think of these things in a very linear direction. We don't know for sure, and I think there are multiple answers. So one answer could be that if you can live inside another organism, you've got some protection from the environment. You might be less likely to be eaten by another organism. You might have a source of nutrients that organism has collected, like in the case of corals, providing nutrients to dinoflagellates. So there might be many reasons that it's useful. But we should also recognize that it might not be a really fair relationship after all. And it might be that the Asgard archaean actually trapped this cell, and it didn't let it out. And perhaps you could argue that it was a bit of a slave or a prisoner. On the one hand, you could say, well, it's been very good for these alpha Proteobacteria because they're now in just about all complex life. So they've actually gone on to live for a long time. Whether that's a fulfilled life is something which we could debate. But I think, again, we need to be careful when we think about these partnerships. They are partnerships, but the different partners might be getting different things out of them, and it might not be balanced. But certainly I think we could think about protection from the environment as being one thing that endosymbionts do actually get to enjoy.
Has each partnership happened only once? So everything with a mitochondria now came from that initial partnership between the Asgardian and the alpha Proteobacterium?
So again, that's a hard question to answer. And for a long time, we assumed that probably it was just once, or at least one successful one that has dominated the rest of the population. So that's the case for the mitochondrion, and it's also been thought to be the case for plastids, for chloroplasts. But the more recent examples that we look at, the more we find out that that's probably not the case. That where we see one type of symbiosis, it looks like that's happened many times, and they've actually all been, have all survived. Increasingly, we're starting to think that this is the case for the chloroplasts. So that Cyanobacterium, that may actually not be a single event. It might have been that multiple events have led to the diversity that we now see. The problem is going further and further back in time, it's harder and harder to decipher all the data that's available now because it's such an ancient thing. At the moment, the assumption is that it probably was a single event for mitochondria that was successful enough that it's led to the populations that exist today. It would have been happening all the time, and other lineages would have gone extinct. But I think it's going to be hard for us to know for sure whether that was a single event that's led to diversity that we see today or not. But looking at the more recent examples of endosymbiosis, we realize that actually it happens a lot, and actually probably multiple events are contributing to modern life.
Yeah. Is there a limit, actually? So can things keep on eating and not eating, but engulfing and partnering, engulfing and partnering, engulfing and partnering? So I think on your side, you got up to, was it 7 cells cooperating with the Dinoflagellate?
And I'm going to make an acknowledgement here. The scenario that I showed you is pretty well supported by evidence, but there's a little bit of uncertainty at that Okrophyte to Dinoflagellate step, and maybe the Cryptophyte. It looks like it went Cryptophyte, Okrophyte to Dinoflagellate, 3 steps, but that's also hard to disentangle fully. And it could have been that there are multiple more events in there that we just don't see anymore, because the process of endosymbiosis often involves keeping what you want and getting rid of all the things you don't need in that relationship. So it means that those endosymbionts often get sort of whittled down to just the things that the cell really wants to keep. And photosynthesis is one of the examples. But that whittling down process, it throws away a lot of other evidence of what else was there. So it might be in that case that there's more. I would say from what we've learned from biology that there's no theoretical limit that I could see, because it seems that every time a cell is able to enter into relationship with a complex cell that might have had itself a complex history. And it's not that all living organisms have gone through those 6 or 7 steps that corals have, but others, may well have gone through more of those steps. So it's again, it's a difficult number to settle on. But I think the lesson is that these partnerships keep happening, so the likelihood is they'll happen more.
Yeah, and this is a good question. So actually, how do you look back across history to study these endosymbioses and when they formed?
So we basically have to. We're stuck without time travel, which time travel would be the perfect tool for studying these, but we don't have that at the moment. So we've got to actually look at what's living now. And for instance, if we look at chloroplasts, the wonderful thing is that chloroplasts have still got some of the DNA from that Cyanobacterium inside them. So there's still some molecular evidence, if you like, and it's very detailed evidence that's in the DNA there that tells us about what that organism looked like. And in fact, a lot of those steps, there's a little bit of DNA that gets left behind or gets maintained that that basically is a history of where that cell came from. So we certainly are using genetic evidence that, as I say, if we look in Dinoflagellates, we can see genetic evidence of everything going right back to the alpha Proteobacterium. Again, the problem is as those symbioses get whittled down, a lot of that evidence has slowly been removed, but there's still enough to look at there. The other thing which we can look at with photosynthetic endosymbiosis is the pigments, the photosynthetic pigments. And you might have noticed that the color, although it started off with Cyanobacteria as a bit more green-like, once we went into the reds and browns, the pigments actually stayed largely the same or very similar. And again, there are other molecular or biochemical signatures that tell us a little bit about the history and where that cell came from. So there's actually quite a wide range of evidence. The problem is the further back in time we go, the more that evidence has started to sort of erode or be lost.
Yeah, I guess it's that remnant piece of DNA that allows you to sort of try and work out that the mitochondrial ancestor, that endosymbiosis event only happened once.
Well, it tells us what the organism was that it came from. What it doesn't tell us is if similar alpha proteobacteria might have been involved in multiple symbioses. So I think it's, you're right, it gives us a good indication that it was perhaps one event or at least one group of organisms that were in those relationships, but it doesn't formally tell us that there weren't two alpha proteobacteria that separately engaged in these new endosymbiotic partnerships.
Yeah.
I think it's always important when we think about what we know or what we think we know, also identify what we pretty sure we don't know for sure, and that way we can keep asking questions.
That is the goal of science, really.
Yeah, absolutely.
Here's a good question. So what is your personal belief for how the evolution of the last eukaryotic ancestor happened? So do you think it was endosymbiosis via some phagocytosis-like process, or a gradual process of organisms living in a biofilm?
So this is a hotly debated topic, and
I'm sensing it might have been a scientist that asked this question, actually.
Yeah, indeed. Look, I'm going to be quite honest and just say that I don't know. I think that there are three, reasonably plausible explanations. The more we learn about Asgard archaea, the more we realise they've got a lot of the capabilities of modern cells that we don't really think about for these simple cells. So it's possible that they could have swallowed up another cell. I think we're getting more and more evidence to suggest that that's possible. The problem is it still was a cell that didn't have much energy, and people would say, well, that suggests that perhaps that's not the way that relationship started, because swallowing things takes a lot of energy. It could have been, we also see that there are some bacteria that can burrow their way into other cells, including other bacteria, so that's also a form of possibility. But I think an equal possibility is that we know, that's right, in biofilms, cells living together in close association, and those associations can just be through physical contact. Cells don't necessarily have to get inside other cells to benefit from their partnerships, but that might have been just so stable that over time one cell just gradually sort of swallowed up the other cell, just sort of merged itself around. I honestly think that all three possibilities have to be considered as contenders, and I don't really have a personal favourite, I think, at the moment.
The person that asked this question is actually an A-level biology student, so hopefully someone who is clearly destined to become a scientist. But on a sort of tangential question, actually, because you're also the head of postgraduate research in our department, and seeing as this is an A-level student who is asking these questions, I wonder, could you just give a bit of information about what a PhD is like in our department? Because hopefully this person will apply to Cambridge University and maybe study with us in the future.
Sure. Look, a PhD or a postgraduate study is all about taking all of the information that has been acquired through studying science, through lectures and reading textbooks, and that's largely what we do in school and then for the first part of undergraduate studies. But what I remember when I was in about my second year of university undergraduate study is you get to the point of knowledge where we're at the threshold, we're at the point where we don't know what comes next. So what we have to do as scientists, and what postgraduate study is all about, is learning how to ask those next questions, how to identify what are the questions that we need answers to, what are the things we don't know. And once you know those things and decide that these are important things to know, to work out how to robustly do experiments and get answers to those questions. So postgraduate study in biochemistry and throughout the world is all about becoming good at identifying questions and then working out how you can answer those questions with good experiments. So there's, it's basically about becoming an independent scientist or independent researcher. And the goal is that once you finish a PhD or something like a PhD, that you'll actually be ready to start your own research program or be on that journey where you can start to direct, what do we need to know in society to look after ourselves, to look after the planet, and how do we get answers to those things? So that's certainly a big part of postgraduate studies. I mean, we also know that a lot of people who do postgraduate studies will not go on to be research scientists. They'll become all sorts of other professions. And again,
science communication being one of them.
Absolutely. And But it's important that you and everybody else that's educated in science has got a good understanding of how science works, not just the outputs of science, which you learn at school, but how science works, because the whole society has to support these efforts for scientific research. So again, there's a lot of professional training and skills to be good communicators, good researchers, to be able to research the literature. You come out with a whole range of skills from a postgraduate degree.
Yeah, I would also just echo that and just say I thoroughly recommend it. My PhD was some of the best years of my life so far. It's a fantastic opportunity.
Yeah, I agree. Mine too.
Well, you did yours in Australia, so
I don't think it would have mattered.
I think we've got time for 2 more questions. So firstly, I just want to ask Latika, did you have anything you want to ask?
Actually, mine is sort of similar to what Ella has asked in the chat there, which is what can be done to prevent coral bleaching from occurring? And like, I suppose another thing is it one of those catch-22 positions that we're in, whereby because of the acidification of the ocean, that we're not going to be able to have as much of the phytoplankton.
Yeah, look, I mean, it's such a difficult problem. I mean, in some respects, we could boil it down to a simple problem. I mean, at the moment, we still don't really in detail understand the nature of that relationship and why it goes wrong. But we do know for sure that if the temperature gets too hot, we see bleaching events, and we're certainly seeing some terrible bleaching events in Australia, but around the world, and it's directly correlated to ocean temperature. So one problem is that we're making the planet hotter. But you also raised that important point too, that CO2 in the atmosphere doesn't just make the planet hotter, it also dissolves in the ocean and makes the ocean more acidic. And the problem with acid is that it dissolves calcium carbonate, which is what corals make their structures out of. And it's not so much that it just dissolves the calcium carbonate, but it makes it harder for corals to build the calcium carbonate in the first place. So we're doing 2 things, or at least the CO2 in the environment is doing 2 things that are damaging this ecosystem. And as I say, the simple answer is we stop emitting CO2, we try to eliminate the CO2 or bring the CO2 levels down. I think on top of that, I mean, and let's just be sort of glum and say we're not going to change those things quickly enough, but there's probably other stresses that we can try to eliminate from the environment as well. We know that nutrient loading runoff from agriculture into marine systems is also probably one of the stresses that contributes to coral bleaching. So these are things that we can control in a more short-term fashion. We know also that corals require healthy fish populations, because in fact the fish are like the farmers in the corals. Some of the fish are eating the algae which would otherwise grow over the corals. We need to be careful and sensible about the fish that we harvest from the oceans as well. So I think there are things that we can do in addition to the big problem, which is CO2, but there are other things that we can do. Again, this is a very interconnected environment, and we need to be managing it as responsibly as we can.
Thank you.
Yeah, thank you.
Did that answer your question? That's okay. Cool. So I think it's time to wrap up. And there's one final question about undergraduate courses at Cambridge. So there isn't a specific biochemistry course at the University of Cambridge. Everyone does natural sciences, and then you take different modules. So in the 2nd year, part 1B, that's when you can first become slightly more specialized in biochemistry with the biochemistry and molecular biology course. It's a fantastic course. I did that course. And then in 3rd year, you can take just biochemistry. And then there's also an optional 4th year in biochemistry that leads to a Master of Natural Sciences degree. All the information on these courses can be found on our website. So that's bioc.cam.ac.uk. So bioc.cam.ac.uk. And if you have any questions, Ross's contact details are on our website, as are our head of undergraduate teaching's contact details. And I'm sure they'll be happy to answer any questions that you have. So with that, I just want to thank Ross for a fantastic talk and a really interesting discussion as well.
No, thanks.
I'd like to thank Latika for helping to. Sorry. I'd like to thank Latika for helping behind the scenes.
No problem.
And finally, I'd just like to thank everyone at home for watching and for taking part in the discussion. So stay safe, and hopefully we'll see you either later this afternoon or later this week for another of our live